A huge hello to all my dear fun beings. Hope you're all doing great. In today's video, we'll be seeing the ABO Blood Grouping System. But before starting, don't forget to hit the subscribe button and click the bell icon next to it. Now, let's get straight to the video. The ABO blood grouping is an example of multiple allele system. So, what are multiple alleles? When a particular gene has more than two alleles, they are referred to as multiple alleles. It is not necessary for all of these alleles to exist in the same individual. They may be existing within the species. So, the A, B or O blood group are determined based on the antigens present on the plasma membrane of the red blood cells. These antigens are mainly of two types, that is, antigen A and antigen B. Apart from this, we also have the RH group of antigens, which we'll be seeing later. The blood also contains antibodies corresponding to these antigens. So let us see this with the help of the table given below. In blood group A, we have the antigen A and antibodies of B. That is, in blood group A, since we do not have the B antigen, we have its antibodies. So in case there is a mixture of blood B in blood group A, then the antibodies present in the blood group A will agglutinate the B red blood cells. Similarly, in blood group B, we have the B antigen and anti-A antibodies. And in blood group AB, we have both the antigens A and B and no antibodies. In blood group O, we have neither A antigen nor the B antigen, but we have both the antibodies of A and B. This is the reason O is called as the universal donor and AB is called the universal acceptor. Now, let us see what are the alleles that are responsible for these antigens. The two alleles IA and IB, that is, capital I superscript A and capital I superscript B, are the dominant alleles, whereas the small I is the recessive allele. The capital IA is responsible for the A blood group and capital IB is responsible for the B blood group. The absence of these two gives small I, which is responsible for the O blood group. So, let us see the table below to understand this concept even better. The genotype IAIA corresponds to the A blood group. The genotype IA and small I corresponds to A. Similarly, the homozygous IB corresponds to B and IB and small I also correspond to B. But when there is a mixture of both, that is, IA and IB, we get the blood group AB. And when we have two small i's, we get the blood group O. So, the antigens are produced by these genes, which we'll be seeing in a short while. Based on this, we can predict the blood group of the parent as well as of the future generation. So, I have posted a question in my Instagram ID, which is crisp.biology. So, please do go and follow me there and try answering the question. I will be posting the answers in a short while. Now, let us have a look at this diagram, which shows us the presence of H antigens and how they are present in all the blood groups irrespective of the presence or absence of the A and B antigen. So, what is this H antigen? The H antigen or H molecule is found in every individual. The ABO antigens are complex oligosaccharides which are present in the form of glycoproteins and glycolipids. So the A antigen is formed by the addition of an N-acetyl galactosamine group to the H molecule. This is done by the GalNAc transferase enzyme, which is produced by the A gene. Similarly, the B antigen is made by adding a galactose chain to the H molecule. This is done by the gal transferase enzyme produced by the B gene. So, here we can see the H antigen and how GalNAc transferase or gal transferase converts it into the A antigen or the B antigen. What happens during blood transfusion? Why do we need to take special care during blood transfusion? It's because we need to make sure that the blood that is to be transfused does not carry the antibodies corresponding to these antigens present in the patient. If there is a cross match, then there will be a possibility of blood agglutination. For this, we will need to know what is agglutination and how the blood grouping is done. The blood grouping studies involves the process of agglutination, which we will be seeing in the next video. But for now, we need to know the donor-recipient compatibility. Again, the positive and negative forms of blood may be dealt with in the next video because it involves the Rh antigens. You may just have a look at this table, which we will be discussing in the next video. So now just go hit the like button and the subscribe for more such videos. Also click the bell icon to be the first one to get your concepts right. And don't forget to follow me on my Instagram, and the handle is given in the description below. Let's meet in another beautiful video. Until then, bye bye.